we studied how microorganisms influence the brain and the development of the brain. After doing that work, I've since started a position as a senior scientist, small biotech company called InVivo Biosystems. It's been understood pretty well, more recently, that the community of microorganisms that lives on us and in us and around us influences biology, or any host's biology.
Before we get started, I have a quick favor. I've been self-funding the Finding Genius podcast for 5 years now. I've done over 3,000 episodes. And as you can see on YouTube, we're up over 1 million views on the channel, which is fantastic. The next thing I really want to push on is to get up to 10,000 subscribers, because once we do, we'll be able to put a donate button, and we'll be able to solicit donations to help keep the podcast running and to also get the Finding Genius Foundation moving along. We have a big project studying anxiety, depression, and PTSD, and working on a product to help people overcome these problems, because I've seen them explode recently after the last 2 years of the whole virus situation. So if you would, please subscribe to the podcast. That would help us tremendously. Give us a thumbs up, and check in the description for Buy Me a Coffee. It's about $5. If you could buy me a coffee, I'd really appreciate it. It would help keep the channel going, and I love coffee. Thank you.
Forget frequently asked questions,
common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius podcast, now part of the Finding Genius Foundation. I have Joseph Bruckner. He was a senior scientist at InVivo Biosystems. We're going to talk about a possible gut-brain connection and social development. So we'll get into more Joseph's bio and do his intro now. So Joseph, thank you for coming.
Thanks for having me.
If you would, tell me a bit about your scientific career and where you're at today, and then we'll get into some questions on your research.
Sure. I suppose I started out in science really being interested in things like parasites and viruses, because creatures like that tell you a lot about kind of basic cell biology and how human biology works, and they're just very interesting. And so I started out interested and did a little bit of work in those fields, and then went to get my PhD in cell biology and found a love for neuroscience. So in graduate school, I suppose became a neuroscientist, and after that I looked to combine my interests both in microorganisms like viruses and bacteria and the nervous system. And I was able to do that in the labs as a postdoctoral researcher in the labs of Judith Eisen and Phil Washburn at the University of Oregon, where we studied how microorganisms influence the brain and the development of the brain. After doing that work, I've since started a position as a senior scientist, small biotech company called InVivo Biosystems.
So what were some of the research questions that you were involved in researching that gave you some insight into the microbiome and social development in people?
Yeah, so we were really interested in, well, I guess I should back up. So it's been understood pretty well, more recently, that the community of microorganisms that lives on us and in us and around us influences biology, or, any host's biology. But especially when it comes to the brain, it's been pretty unclear how exactly that works, how microorganisms communicate with the brain, especially during early development. And so that's the basic question that we set out to answer. It is to reveal some of the, what's often called mechanism, which is really just more detail into how microbes send signals to the brain to influence how it develops.
Okay, so what models did you use? Humans, mice, and what kind of experimentation was done?
Yeah, that's a great question. So all of our studies were carried out in a model vertebrate called zebrafish. So it's a little aquarium fish that you've probably seen at your local pet store, but they're one of the most popular model organisms used for research around the world. I can get into why that is if you'd like, but there's a lot of advantages of using zebrafish to answer kind of fundamental questions like this.
Okay, so you use zebrafish. What kind of experimentation was done, and what did you find out that was interesting to you?
Yeah, so there's a number of things, but our basic the manipulation that we started with is that we took fish and we made them germ free. So we removed all of their normal microbes very early in life, and then we looked to see how that affected both their behavior and their brain development. So one of our early findings was that when you do that manipulation, that the fish don't do their typical social behavior the way that they do if they have the microbes. And then kind of investigating how, or why that's the case, we discovered that it's because those microbes are required for neurons in the fish's brain that normally regulate social behavior to develop properly.
Okay, so the microbiome was altering how their nervous system and their brain was literally growing in vitro there before they were born, or at what point did you study the zebrafish?
Yeah, that's a good question. So early in development. So one advantage of fish as a model is that, mama fish lays them as eggs, right? And all of the development happens outside of mom. Whereas if you're trying to do studies like this in a human or in a mouse, a lot of that early brain development is happening in utero, and it's very difficult to study. So manipulating the microbiome from the minute that the eggs are laid, so those fish are never exposed to their normal microbe. And then the developmental effects that we were observing in the brain, those are things that were happening within the first week of the fish's development. So that would be, equivalent to something like a mouse or a toddler. It's hard to make the kind of extrapolate fish development exactly to a human. Yeah. Early development is the point.
Right, and the big contrast is that, again, in human development, the baby is inside the mom until it's born. But with fish, as soon as they lay the egg, that's it. There's not going to be a connection, most likely, between the fish and the egg.
Right, exactly.
Does the fish pass anything to the eggs? I mean, does it nudge them with its mouth? Does it appear to provide any does it try to mouth feed, and maybe some of that material goes into the egg? Yeah, I don't know. I mean, maybe it's a crazy thing to say, but
no, it's a good question. I mean, there is kind of the transfer. I guess for our purposes, what's important is how mom transferring the microbiome in some way, right? Like is she conveying either microbes or, like you're saying, nutrients to the offspring? It is kind of a similar setup as it is in mammals, because this isn't my research, but other research has found that a lot of that transfer of the microbes happens from mom. So, as the egg is laid, that egg is coated in microbes from mom. Mom is still swimming around in the water with the eggs. So, that water presumably has its own microbial community that the eggs are exposed to. And
what about inside the egg? Has anyone aspirated some material to look for microbes that live inside the egg during development?
Yeah, that's a really good question. It's, again, not my research, but my understanding is that inside the egg is thought to be almost entirely sterile. So the fish don't get exposed to those microbes until they hatch. So there's like a couple days where that's, kind of equivalent to a human, right? The human doesn't get exposed to microbes until it's born, where it's in that sterile environment, and then it hatches and it starts to be exposed to microbes and get colonized by those microbes, at which point they can be, influencing its development. There are other ways that the microbes can influence development, via mom, aside from that, but that's what the primary effect we're looking at in this work.
Okay, so what was figured out? Anything, or this was just all hypothesis? And was any data able to be gathered, or what happened?
Yeah, so we found that. Do you mean in regards to how those microbes influence brain development?
Yeah, what kind of microbes were there?
Oh yeah. So
species or strains. What was observed? Did it correlate with the mother's microbiome?
Yeah, we didn't directly address that. There was a small part of the study that kind of addresses that. So the problem is that the initial manipulation that we did, that I was talking about, where we took away all the microbes and we looked to see what happens in the brain, right? That's kind of the fundamental question. What is the microbiome as a whole required for certain things in brain development. But that a germ-free animal doesn't exist out in nature. There's no, germ-free kid running around, or there's no germ-free fish swimming around in a stream. So it's kind of an artificial manipulation, but it's useful for telling us what are the general features of development that are required, for which the whole microbiota is required. We then took those germ-free fish and we added back a select few species or strains, from the whole community to see if they could kind of restore the deficits in those germ-free fish. And it looks like so far all of the strains that we have tried do mostly restore the features of brain development that we were looking at. We haven't done a ton of work in that area.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
How do you even know that the eggs are sterile, or that the zebrafish themselves are truly sterile and other bacteria didn't take up residence temporarily once you clean them out of their existing microbiomes?
That's a good question. We have a pretty thorough protocol that the larger group, beyond just our lab, a lot of different labs have worked for years on kind of developing a method for not only making those fish germ-free to start with, but keeping them in a sterile environment for a while. So we use a variety of monitoring techniques to hopefully detect any bacteria that show up after the fact and kind of control the ones that we're adding back as well.
Okay.
One thing.
So again, what was seen? Did the eggs develop differently? I mean, so before they were laid, the zebrafish were made supposedly germ-free, but did you do before and after?
Yeah, that happens right after they're laid. It's very easy to
what about before they're laid? Did you try any of that and then compare the eggs and the development and all that in the hatch rate?
I'm not sure I understand what you mean. So before they're laid, they're in mom.
Right, but did anyone try to, I don't know if you can give antibiotics to zebrafish or how you'd make them germ-free, but were any done before they laid, or were some done after they laid, just to see if there's a difference?
Yeah, no, I understand what you mean. That's an experiment that's actually pretty hard to do in fish. It's been done a little bit in mice. So those experiments typically are aimed addressing or separating what are the effects of, mom's microbiome versus the effect of the offspring's microbiome. Our study was entirely focused on the offspring, but that work like that is being done in mice.
Okay, so what was observed? What was noticed in this experiment?
So previous work in our lab had identified this population of cells in a part of the zebrafish brain that we already knew were important for social behavior. So when we make the fish germ-free, they don't do social behavior right. And we had this population of cells that we knew were important for social behavior, so we looked at those in the germ-free fish. And one of our first findings was that those neurons, they don't appear to wire up the way they normally do when the microbes aren't there. So, in kind of fundamental neuroscience, the idea is that, how neurons wire up with each other determines how the whole circuit works and enables the behavior to happen. So
maybe I didn't hear properly. So the fish were sterilized after they had laid the eggs, but it somehow affected the eggs? I'm not clear. I apologize.
Yeah, it's just a generational thing. So the eggs were sterile, were made germ-free, but we've removed mom from the equation. So
how did you make the eggs germ-free?
Yeah.
What was the protocol?
It's kind of a complex protocol, but it basically just involves using chemicals like bleach and iodine to kind of sterilize the surface of the egg, because like I was saying, they're sterile inside there. So we kill the bacteria that are there with things like bleach and iodine, and then also through a lot of dilution, remove them while they're still in the egg, and then we can keep them in a sterile environment.
Okay, so you sterilize the outside of the egg, and then so in the act of hatching, do the eggs take on what would normally be on the outside coating of the egg as their microbiome, and then also the ambient, water around them?
Yep, exactly. And so if the microbes aren't there, then those fish won't get colonized.
So what's the difference between the microbes that are associated with the egg surface versus in the free water around them? Have they been characterized, and are they different?
Yeah, that's a fascinating question that is being worked on by a number of laboratories. And I think my understanding of where we're at on that is that it's obvious there is a difference, but it hasn't been fully characterized. An active area of research.
Any guess as to the role of the microbes on the egg surface? Are they trading nutrients with, let's say, microbes in the water around them? And if so, do they build up or change the nature of the outer membrane of the egg?
Yeah, I don't think that's really understood. I'm not a microbiologist by training, but my understanding is that, it's all about kind of microenvironments, and that bacteria will be specialized for those particular microenvironments. And that then affects how they get into the host, right? So those bacteria have evolved with that host for eons, right? So they're very suited to live in those particular environments, including inside the host.
Okay, that makes sense. So at what point were they chemically sanitized, the eggs? Like, how long is their development in the egg, and what day was it done? Like right away or at the end? Or was there any variation in that?
Yeah, that's a good question. So that's done right away, like literally within a few hours of when they're laid. They then develop inside their egg for 3 to 4 days before they hatch. And then their brain is developing all throughout that period from day 3 days onward, from a few days onward. And then by the time they're a couple weeks old, they start to be social.
Okay. But how long is this gestation period that they're in the egg until they hatch?
About 3 to 4 days.
Okay. All right. Again, was there any experimentation where it was done on the 3rd day or the 4th day, or was it all just done on the 1st day and it was enough to experiment on? Too many variables otherwise.
Yeah, but it's a really good question. The short answer is too many variables otherwise, but in further experiments, we'd really like to know if there is kind of an earlier critical period. Like, does it matter if you're missing the microbes, only at day 3 or at day 4 or at day 5? What's kind of that specific period within that early window that those microbes are required? And we haven't done those experiments yet.
Yeah, so their fish looked normal, phenotypically normal. It was just their social behavior that was altered, but in every other way that you guys looked at them, they appeared to be healthy. You couldn't tell that there was anything going on with them. Is that what you're saying?
Yeah, that's right. So all the measures that we looked at, they're phenotypically normal. So they swim around. They're not smaller than their siblings. They appear to be able to see just fine. It's just that their social behavior is altered.
Okay, so when you say social behavior is altered, what did you observe? Did the behavior aberrations happen right when they were spawned or hatched, or did it get worse later on? Like, what did you observe?
Yeah, that's a good question. So typically, the fish don't start to exhibit any social behavior until around 12 or 14 days. So a few weeks of life. And so we measured the social behavior at that age. And what they normally do is they prefer to, if you take 2 fish and you separate them, except for a transparent divider, they prefer to be really close to each other. And they'll actually swim along the divider. We think maybe trying to get a good look at the friend, and generally spend a lot of time close to that other fish. And that's the type of behavior that was altered when they're raised without their microbiota, is that they no longer seem to care. They no longer spent much time close to the other fish.
Interesting. Has anyone looked at the microbiome variations across different zebrafish? That, it'll be interesting if you looked at the microbiomes of zebrafish that hadn't associated yet, and then let them associate for, I don't know, an hour or something like that, or a day in a tank, and then look at their microbiomes and see if they're influencing each other's microbiomes, or what what kind of resources or things may be traded that go along with this social behavior? Why does the microbiome seem to be driving this social behavior?
Yeah.
What benefit does it get,
Yeah, that's a really good question. There are a number of labs who are looking into that. Our focus was more on the host side of things. So, what features of the host are the microbes influencing? But it's an excellent question and a really active area of research looking into why does that matter for the microbes, right? You could hypothesize that promoting social behavior would be really great for bacteria, right? Because it could help them spread from fish to fish and things like that. That's definitely a hot area of research.
Yeah, if they had the same microbiome or similar, then that wouldn't maybe make sense. But if it was different and the social interactions allowed the differing microbiomes to, let's say, average out to be healthier or to interact and trade molecules that were somehow important to the fish, I guess a lot of questions surrounding it.
Yeah.
What happened over time to the antisocial fish? Did they live as long? Did they just never mate? Like what happens to them?
Yeah, that's a good question. They still appear to be relatively normal in a lot of other effects. We didn't follow them for their whole lives, for example. That wasn't the focus of the research, but they certainly maintain relatively normal health for as long as we observed them.
Were males and females affected differently? Did females end up laying eggs, or were they just not made at all? I mean, what other things jump out at you from their behavior?
Yeah, we didn't look at that. We focused a lot, again, on kind of what's happening inside the brain. One big question we wanted to ask is, like I said, we found that these neurons in their brain that are really important for social behavior, those neurons weren't wiring properly. Well, the brain is in the head. The microbes they're all over the fish, but largely in the gut, on the skin. So how are those separate pieces communicating in the gut, telling the brain what to do? And our experiments, it looks like a lot of that has to do with the brain's immune system, that the microbes are kind of influencing special immune cells that are then talking to those neurons as they develop and then influencing the social behavior.
Okay. So what do you think will be some of the implications of the research? What's its goal and where is it going?
Yeah, that's a great question. The big goal of the research is to shed light, like I said, on how these things work mechanistically, right? How this communication happens. And so our hope is that if we learn more and more details, like what we've discovered about how exactly microbes are influencing development, then that gives us all sorts of new areas, both in the microbes and in the host, that could be targeted by new therapeutics. So it's understood now that microbes are implicated in a lot of different disorders, including many brain disorders. And so if we come up with more clues as to how that works, then these potential treatments could be really broadly effective across many different disorders that have so far been really hard to address.
Well, what would be a treatment? Like, what would you do? Is certain people maybe that have risk factors would, undergo some sort of microbial change, maybe given antibiotics or some other protocol that would then hopefully change their offspring's ability to live properly and socialize?
Yeah, that's the idea. We're a long ways off still from making those actual treatments, but I think the hope is that we could get away from things that are more of a sledgehammer to kind of more fine tuning those interactions between the microbe and the host. For example, there's been some interesting clinical trials with children with autism spectrum disorder, where the intervention that the clinicians are doing is called a fecal microbiota transplant, which you may have heard of. And essentially, that just involves transplanting the microbiome, so giving them the microbiome of a healthy person. But like I said, that's kind of a sledgehammer. right? You're changing their whole microbiome. And those studies have been promising, but limited. And so our hope is that if we understand more about how the actual interactions work, instead of changing the whole microbiome, we could kind of, pull the levers and twist the knobs a lot more gently to tune those interactions, the host and its microbes.
Okay. Well, very good. What's the best place for people to learn more about the research? I know you've moved on, but for people listening, how can they find out more about what the lab's still doing and, other ancillary topics in this area?
Yeah, that's a great question. I would say through the University of Oregon's website, this work was done in part as part of a larger group at the University of Oregon called the Meta Center. So it's the microbial, I might get this wrong, microbial ecology and theory of animals, I think, but double check me on that.
Okay.
The Meta Center hosts a lot of the host microbe interaction research at the University of Oregon, and there's a lot of fascinating research being done in that group. So I would definitely encourage people to check out the Meta Center's website, and which will have links to the individual research groups and their research.
Okay, well, very good. Joseph, thanks so much for coming on the podcast. And, it's rare is when people are not directly working in their area at that moment that I interview them. You're one of the few that's brave enough to be willing to talk about it. So I really appreciate you doing this, by the way.
Absolutely. I think it's, I really enjoyed doing the work, and I think it's a fascinating field and really important work. So I'm always happy to talk about it.
Excellent.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.